Welcome to the CJD Foundation Virtual Conference. We thank our partners, the Centers for Disease Control and Prevention, and the National Prion Disease Pathology Surveillance Center, and our sponsors, Charles River Labs and Ionis Pharmaceuticals. Tonight we feature one of 3 panels showing updates from the 8 scientists who were awarded grants by the CJD Foundation in January 2021. Everyone who donates to the CJD Foundation and to Strides for CJD has a part in funding these projects. We also send special thanks to the families who have donated memorial research grants to support these important research studies. Together, you are helping the prion disease community unlock answers to some pressing questions about prion disease. To learn more about family memorial grants or about Strides for CJD, visit cjdfoundation.org or call our office. During tonight's panel presentation, please enter your questions as they occur to you. The panel will answer as many as possible following the presentations, focusing on tonight's topics. If your question is not answered, feel free to email it to us at help@cjdfoundation.org. For information on topics not covered tonight, please visit our conference website to view previously recorded presentations. And now I'm pleased to introduce our moderator, Professor Richard Knight. Professor Knight is a clinical neurologist who has served in the UK National CJD Research and Surveillance Unit. He's a professional fellow in clinical neurology at the Centre for Clinical Brain Sciences, University of Edinburgh. He has served as chair of the UK National CJD Support Network Management Committee. We're grateful that Professor Knight joins us each year for the CJD Foundation Conference in Washington, D.C. He's a past keynote speaker and also served as panel moderator at our 2020 virtual conference. He will introduce tonight's speakers and will moderate the Q&A panel following the presentations.
I'm very happy to be here at the CJD Foundation Virtual Conference, Panel 3, which is where you, the families, have the opportunity to hear about the research that you funded via the Foundation. And we have 3 speakers today who are going to really talk about therapeutic treatment issues. They'll present their research in a way that I hope you'll understand, and thereby you'll see how valuable your financial support is. You will, of course, have the opportunity to ask questions, and I will introduce the speakers in turn and try to put their research into a sort of brief context. So, first speaker is Dr. Biasini, an associate professor at the University of Trento. He has a long background in prion disease research and has a particular interest in the development of treatment for these diseases. As a broad context, in prion disease, a normal protein that we all make and have, the cellular prion protein, designated PRPC, is converted to an abnormal form designated PRPSC. And one treatment approach is to reduce the normal protein so there is less of it to convert to an abnormal form. And Dr. Biasini is interested in methods of identifying chemicals that might reduce the normal protein and then developing these into drugs that could be then evaluated in humans. So without further ado, I will ask Dr. Biasini to give the talk, Pharmacological Degraders for the Cellular Prion Protein.
Okay, so thank you very much for inviting me to present our project that was supported by the CJD Foundation this year. And I will start by thanking all the families and I will say the whole CJD Foundation for supporting our research throughout the years. A little bit of background of where we are. We are in the northeastern part of Italy, nested inside the UNESCO World Heritage Dolomites, these fabulous mountains. Our center is characterized by the definition of cross-disciplinarity, because in reality, what we have is lots of different techniques and lots of different expertises brought by the faculties that come together to make a true cross-disciplinary science that try to break the barriers between disciplines. And I hope that what I will present today will provide an example of this concept. So my lab has been active now for several years on a single protein. I keep saying that I became monomaniac in my career because the reality is that I just study one single protein. That is, this protein probably, quite familiar with all the audience that are hearing us today. The name is the cellular prion protein. This is a protein that usually resides outside cells, in particular nerve cells, on the surface of these cells. Unfortunately, we still don't fully understand what this protein does in the brain, but we actually understand something, let's say, dark about this protein, its ability to eventually change its form, something that in biochemistry we call conformation. Well, this protein is able to basically change its form to become something else, something actually toxic and dangerous, something that then polymerizes and makes this sort of structure that you can see here on the right of the slide that we call amyloid fibrils. And these amyloid fibrils eventually accumulate in the brain of patients affected by prion diseases and, of course, create a very devastating kind of phenotype that we all know. So historically, in the prion field, people have been working on the idea of trying to clear up these amyloids from the brain of patients. Eventually, this concept got a little bit old-fashioned when we realized that this was very difficult, if not impossible, to do. In parallel, another concept started to emerge. That is the idea of actually targeting the normal form of the protein, so the cellular prion protein that I introduced a few seconds ago, the protein that we all have on the surface of our nerve cells. Targeting this protein pharmacologically in various different ways would actually represent a way, an opportunity for blocking the formation of those toxic amyloids at the very beginning, because we will actually stop that misfolding process at the very beginning. And without taking too long, there is clear evidence in the literature that we can actually block the expression or actually block the function or both of these protein without damaging, without creating any damage to the nerve tissue, eventually to the human being receiving the treatment. In the literature, especially in the data collected in laboratory animals, we know very clearly that these kind of concept of blocking the expression, so reducing the loads, of abrogating completely the expression of this protein in animal models, have a tremendous impact, beneficial impact on the disease. Just to make an example, if we create mice that are completely knockout, meaning that they don't express at all the cellular prion protein, they are 100% resistant to any prion disease. And the same will very much be true in human. In fact, this is actually an image that we published with a few colleagues a few months ago to try to rationalize ways that we could try to explore in order to obtain this effect, meaning reducing the loads of this normal protein in the brain of patients. And one immediate intuitive way is pursued by Ionis Pharmaceutical, which is trying to develop so-called ASOs, or antisense oligonucleotides. These are compounds, are non-canonical drugs that actually impair the normal production of the protein at the very beginning again, and eventually then reducing the loads of the protein in the brain. And this is definitely a very promising approach. What my lab is trying to do is something that we consider complementary to what Ionis is doing. We want eventually to try to answer to this question. Is there a way to selectively suppress PRPC with canonical drugs? By canonical, I mean small molecules, so drugs that, like many others that are in the clinics for treating many diseases. And Okay, in order to explain what we're trying to do, I will start very easily to recapitulate how a protein is made in a cell, in whatever cell you may imagine. In fact, what you see here is called the central dogma of biology, and it's basically transmission of information, an information encoded inside the DNA that then gets transcribed into a molecule that we call RNA. This RNA is processed to make a messenger RNA, and the word message means that then this molecule travels to other kind of structures in the cells that are called ribosomes, and thanks to the information encoded into the mRNA, the ribosomes basically print proteins by assembling one amino acid at a time. And the endpoint, the result of this translation of the information encoded into the mRNA, is basically a long chain of amino acids, a unique chain, unique for each protein. Now, there is another process, very important here. This chain of amino acids that you can see here then fold on each other, or each amino acid on each other, to specify that form of the protein that I referred to as, a few slides ago, and then makes the final form of the protein, for which, thanks to which the protein then exerts its biological function. Now, we are particularly interested in this process, this folding process. It turned out that this process is very complicated, because you can imagine that some protein may actually be made by, several hundred amino acids. Our protein, the cellular prion protein, the subject of this talk and my research, is actually roughly 200 amino acids. Now, each amino acid has its own property, chemical physical properties, and the combination of these properties then result in the final form, meaning the folding process and the final form of the protein. There is a paradox in the scientific community called the Levinthal's paradox, that basically if you take whatever kind of biologically relevant protein and you try to reconstruct all the movements that the amino acids should make to then lead to the final form of the protein, then you will come out with an astronomical number of possible conformations, meaning possible movements that then result in the final protein. So this is just to summarize, it's a very complex kind of field of science to understand how proteins really reach their final conformation. One help actually definitely comes from recent emerging computational technologies. And in fact, today it's possible, thanks to large supercomputers, to simulate the entire process of folding for very small proteins. Proteins that are, unfortunately, in the vast majority of cases, are not biologically relevant. To make an example, to simulate the entire folding process of the prion protein, it will take us 1 per 10 to the 14 microseconds, which are basically hundreds to thousands of years. So feasible, but practically unfeasible. Now, what I discovered once I joined the University of Trento is that sometimes it's much better to have a good colleague than a large supercomputer. In fact, this colleague of mine in the Department of Physics, Pietro Faccioli, actually invented a way of simulating folding of proteins in a very short time. So in the range of weeks to months. And then the result is basically this video that you can see here. That is the entire process of folding of the prion protein. This is the very first time that someone can reconstruct this entire process that you can see as a movie. Now, just for reason of time, I will get to the point. Along the process of folding of the prion protein, we identified an intermediate species, meaning a form of the protein that is transient, but still existing for a very short time, but existing, like a form of the protein that precedes the final state that we call the native state, so that the final form of the protein. Now, one idea then was that if we are able to target this transient intermediate state along the folding pathway of the protein with a drug, then we could actually stop the production of the protein, because we know that the quality control that every cell has will recognize that not completely finalized, let's say, form of the protein as an improper product, and then trash it out. So we tested this hypothesis, but then applying kind of more canonical computational and experimental techniques that are common in pharmacology. In particular, here we try to target a specific part of this intermediate form of the prion protein with lots and lots of drugs. We came out with 31 restricted molecules, small molecules binding in that pocket. And likely for us, one of these molecules, in reality 4, but I'm presenting data just for 1 today, one of these molecules was very effective in doing what we predicted. What you can see here, this black kind of stain signals, is nothing but the cellular prion protein that we detect with an antibody. And as you can see from left to right, the erasing amount of our compound called SM875 is corresponding to a difference in the signal of the prion protein, and eventually a drastic reduction, something that the graph below exemplify very well. So this protein is the first degrader, so a molecule that can block the folding and then leads the protein to degradation that we know so far for the prion protein. Now, few other data. We know, and I'm just gonna summarize lots of data in few slides. We know that this compound is very specific for PRPC because we tried it on different proteins that are very different from PRP, but share many different, let's say, features, like the way that these proteins are expressed is very similar to PRPC. And to make a long story short, the compound does nothing to these non-relevant proteins. To summarize, again, a great deal of data, what we found was that our molecule is truly impacting the formation, the first, let's say, production of the protein during the folding process, so that the protein actually never reaches its usual location, which is the surface of the cell. And thanks to this property, then, our compound, incubated together with cells that are replicating prions, is able to really drastically lower the amount of prions that are replicating in these cells. So what we do now with this very promising compound that is acting in a completely novel mechanism, at this point, we try to go along with the classical, let's say, drug discovery pipeline, meaning that now that we collect direct information on the properties of these compounds in the lab, especially in cell cultured in our lab, now we're trying to make it better in order to be able to provide this compound to organism, mice first, and experimental models that we can handle in the lab, and then, if we're lucky, also translate that information to human being. So now we are exactly at this point, and thanks to the support of the CJD Foundation, we are making lots and lots of molecules that are very similar to the SM875, which we call the precursor molecule, but we test all the time. Whenever we make new molecules, we test their properties. And if we're lucky, we improve the properties, and at the same time, look for properties that are ideal for a compound to be provided in vivo, so in real organism. And I mean, I don't want to enter into the details, but we are reaching molecules. We already have identified molecules that are slightly better or even much better than the one that we initially identified, so the precursor molecule. And this is just recapitulating what we want to do. We want to fully explore the chemistry of this compound, and then also study what is called the pharmacokinetics, meaning how this compound may interact with a living organism, and then test its therapeutic potentials directly in animal models. At the end of this, feedback loop, if we're lucky, we come out with a molecule that is administrable in vivo in human beings, and at that point, we will enter, clinical phases. We're still far away from that, but we believe this is a very good, promising starting point. And I will just finish by thanking a large cohort of collaborators here from, many different places around the world. And of course, our funding entities, the Teleton Foundation, that's basically sponsorized my lab, and of course, the CJD Foundation that brought me here today, and all the audience for the attention.
So Dr. Dusky is our second speaker and works at the University of Medina and Reggio Emilia. He has an interest in developing methods of delivery of compounds to the brain. And the brain is separated to some degree from the body. It is protected to some degree by things that happen in the body by something called the blood-brain barrier. And this means that drugs that are taken orally or by injection might not in themselves be able to actually get into the brain. Now, prion diseases obviously affect the brain, and while a group of chemicals called porphyrins have potential useful effects on prion disease mechanisms, these chemicals cannot normally cross the blood-brain barrier. And Dr. Dusky is interested in developing a suitable transport system to overcome this. So I'll ask Dr. Dusky to give his talk on optimization of nanoparticle-mediated brain delivery of a tetracationic porphyrin with proven antiprion properties.
So thank you for the introduction. I'm Jason Dusky. Today we'll be talking about our nanoparticle-based system with a novel drug that we created in order to fight prions disease. A little bit more about me, since I am a born American, but today I'm working in the University of Modena in Italy. I'd like to talk about our university a little bit. Our department's a little bit smaller than what we're used to in the United States, with only about 50 principal investigators and 30 laboratories, but we all focus on nanobiomaterials and nanotherapeutics in order to create new drug systems and new therapeutic-based systems for hard-to-treat diseases. I won't go too much into the details of the disease, since I'm pretty sure most of you know as much about it as I do, but I would like to point out one important fact, that this disease is caused by a protein, which is the prion protein C, which is a natural protein that we all have in our bodies that's soluble and can be degraded in our system, so therefore it's not toxic. However, a change in its 3D structure can lead to it to group together with other proteins and become insoluble, and therefore the body can no longer degrade it, and this is what causes the toxicity in the system, and this will be important for the drug that I'll be talking about today. And this can happen either through being introduced through one of the disease state proteins or genetic mutations, or through a sporadic change. So in the literature, there's a lot of different molecules that have been tested in order to try to combat this change between the two proteins. One of the main ones is this iron-containing one in the middle, and this is a porphyrin, which we'll be talking about throughout the talk. It's capable of binding to the PRP-C or PRP-SC proteins in order to block the transition from the safe protein to the disease state protein. Now, for us as scientists, we're able to calculate how much of this effect happens by looking at the amount of protein in cells. So in this graph here, you can see in normal cells where there's a lot of the diseased protein, we can see a large band, and then by adding the drug, we can see that this band disappears, meaning that we're degrading and removing that toxic protein. Now, we're trying to create new, novel, and better drugs, and so in our collaboration, we've created a new drug based on this first iron-containing molecule, which unfortunately I can't show the structure of due to confidentiality agreements, but we can compare them directly based on the results. So in the ability to knock down the disease state protein, we can see that our protein at the same concentration of the iron-containing protein actually has a larger knockdown effect compared to that, which is the prominent drug in the literature. More importantly, or more interestingly, is that this iron-containing molecule does not affect the safe, natural prion protein that we all have in our systems. Instead, our drug actually does knock it down. And you might be asking why we would want to destroy or remove a protein that we all have that's safe and that's natural. But scientists have actually discovered that even the natural protein is not critical to being healthy. So there's animal models that show that even without the natural protein, that they are healthy and there's no problems. And this is very important for us for a few reasons. One is because testing the disease state protein is actually very dangerous because we're putting ourselves into contact with this disease state protein. This allows us for safer testing because we don't have to use disease state cell models. We can use just the natural cell models, but it also leads to cheaper testing. And it also leads to the possibility of, it also leads the ability to stop the disease before it happens, so a preventative effect. However, this drug that we created, when injected in the mice models, is cleared from the blood rapidly, and so it goes directly into the liver and is removed, and therefore has no effect, and it cannot get to the brain, which makes sense because our brain is naturally designed to protect us from anything that's trying to get from the blood and into the brain, which is important because it's our control system. And this is because it can block 80% of all drugs that go into the bloodstream. So for that, we have to look towards nanomedicine-based approaches, which there's lots of different types of nano-based approaches. Maybe some of you have been keeping up with the COVID vaccines, which are lipid-based nanocarriers. But there's lots of different base nanocarriers, and what they are nanoparticles that can be ranged from about 10 nanometers up to 100 or up to about 500 nanometers. And this puts us in the range of nanoparticles that would be about the size of a small flu virus or around a bacteria. And these are very important because they can allow us to increase the compatibility of certain drugs with the human body. And it also allows us to target these drugs to different parts of the body, such as the brain. By doing this, we can lower side effects, and we can also control the treatment where the nanoparticles can release the drug over long periods of time, meaning it's not just a one-shot short-term treatment, but it can also be long-term, decreasing the number of doses and how often we would have to treat for any disease state. In our lab, we specifically use a polymer called PLGA to make our nanoparticles, which is polylactic coglycolic acid. It's a safe FDA-approved polymer. It's fairly inexpensive and creates these nice round particles that can be loaded with drugs. More importantly, we've shown that by decorating the surface of these nanoparticles, we can deliver it to the brain at about 10% of dose, which is about 100 times more than what you would expect any drug to be passing into the brain. And we've shown this already in another disease state model. So now that we have a new drug that is better than that works better than the one in the literature, and a way that should be able to transport it to the brain, we wanted to optimize these nanoparticles to carry our drug to the brain. Not to get too specific, but by loading the drug into the nanoparticles, we can see that we can maintain a desired size. They are uniform and reproducible, so then we don't have to worry too much about different batches having different effects. And by optimizing the amount of drug, we can load high amounts of drug into the nanoparticles and not waste a lot of nanoparticles, making it less costly to produce these nanoparticles. So just because we created nanoparticles does not mean that they'll work, because sometimes nanoparticles can affect the efficacy of a new drug. So we tested the drug by itself compared to the drug inside the nanoparticles at similar doses. And surprisingly, we actually found that the drug, when encapsulated in the nanoparticles, has a higher effect, which you can see on the left side, the left green box, compared to the right green box, which would be the drug alone. So inside the nanoparticles, we actually have a stronger effect of the drug. As I said, we can also control the release. So with the drug alone, or with the nanoparticles with the drug, we see that within 1 hour of dosing, we have completely destroyed the prion protein. However, with the drug itself, this protein is synthesized in the cells within 24 hours. But with our nanoparticles, we don't see the reemergence for about 48 hours. So that means there's also a longer lasting effect. And just to clarify that this is not an effect of the nanoparticles themselves, because the nanoparticles have no effect if the drug is not loaded into them. By delivering these nanoparticles, we can see that they can enter into cells, which are the little red dots surrounding the blue cell marker, within 12 hours. So this means that by dosing these, we can get them into the cells within 12 hours. The effect would occur within 1 hour, and then it would last for up to 48 hours. However, unexpectedly, when we dosed these into mice again, it didn't go into the brain, which is strange because we've already shown these nanoparticles to go into the brain, and we didn't understand exactly what was occurring. So what went wrong? Normally, when we look at these nanoparticles, we take our drug and the polymer, which is decorated with the peptide shown here to deliver it to the brain, and we see that the drug goes inside the nanoparticles, and therefore the peptide on the outside can take the nanoparticles and deliver it to the brain. However, with this specific molecule, there were interactions that we were not expecting, where these 3 amino acids of the peptide can directly interact with the molecule, which we don't see very often. This basically caused the molecule to coat the outside of the nanoparticles instead of the inside, blocking our peptides, which should transport it to the brain. So what can we do? By modifying that peptide, we can now make new peptides that can also have brain targeting, and we can affect them, we can change their structures in order to lower the interaction. So as you can see with the blue box, we only have 1 amino acid there, which is similar to the ones which had interactions in the last one, which means more of the peptide will still be available without interacting with the molecule and being blocked. More importantly, we added a spacer, another safe FDA-approved spacer, in order to lengthen the peptide in order to remove it from the surface of the nanoparticles. So therefore, even if our molecule binds the nanoparticle surface, the targeting peptide is removed from the surface and therefore will be outside the range of the drug, allowing it to still have targeting effects. Moreover, just to keep our options open, we're modifying this peptide even further to have absolutely no interaction with the peptide. And we're also looking at 2 antibodies that will target in different ways that should have less interaction with the molecules. So in the end, we've created a new molecule which can degrade both the normal and the disease state prion protein. It works better when encapsulated in nanoparticles, giving us even higher therapeutic potentials. And we've elucidated all the interactions which had the molecule binding to our targeting peptides and blocking their ability to target to the brain. So now that we have these new peptides designed with less interaction, we're synthesizing them and testing them for their interaction, as well as with the antibodies. And mouse model testing is now underway to deliver this new molecule to the brain and knock down the prion proteins in order to have a therapeutic treatment. With that, I would like to thank everybody who has helped fund us and all of the family and friends who have been affected by this terrible disease. And lastly, I would like to thank my group and all of our collaborators, which have allowed us to create such a promising therapeutic treatment with the new molecule and our nanoparticle systems. Thank you for watching, and I would gladly take any questions.
So our third speaker is Dr. Villa, who works at the University of Alberta, Edmonton. And he has an interest in the structure of prion proteins and how understanding the structure might aid the development of treatments. Antibodies are molecules that are directed at foreign threats to the body, and all of us will have heard a great deal recently about the generation of antibodies to certain parts of the COVID-19 virus, for example. But antibodies can be made outside the body and then administered to people to act against some form of bad protein in their body. And they have been developed, antibodies, against the normal prion protein, PRPC, as a suggested treatment in order to reduce the amount of normal PRP. But Dr. Villa has been interested in developing antibodies against the actual abnormal disease-related prion protein, PRPSC, and there are potential advantages in this approach. So I'll ask Dr. Villa to give his talk of evaluating possible immunotherapy in prion-infected transgenic mice expressing human PRP.
Hello, my name is Holger, and I'm here today to present you the results, so far as we have them, of our project titled Evaluating Passive Immunotherapy in Prion-Infected Transgenic Mice Expressing Human PRP. Now, this project, of course, is funded by the CJD Foundation, and I show here the long list of the various grants and the people who contributed to these grants who made it possible for us to do this research. This list is so long that I don't want to read it out individually. I just wanted to give you my heartfelt thanks for providing us with these funds so that we can do the research, because without these funds, it wouldn't be possible for us to do the work that I'm going to present to you in a moment. Now, I also have to disclose that we did file for patent application for the things I'm going to talk about. We would like to commercialize these in either antigens, vaccines, or antibodies, but no action has been taken on that. And I also have to acknowledge prior funding from the Canadian Institute of Health, the Alberta Livestock and Meat Agency, the Alberta Prion Research Institute, and also the CJD Foundation for a vaccine project, which is still running a little bit at this time. And I'm going to show you this data and mention it in particular and how this one helps for the current project that I'm presenting today. So, antibodies as therapy. This is what this project really is about. And obviously, we all heard about antibodies as therapy for COVID. So this is a very common one. What I'm going to show here in the slide is a schematic, one of these antibodies. These are rather complex molecules. In fact, each one of these antibodies is actually 4 proteins, because there's 2 heavy chains, which are schematically drawn here in light blue, and 2 light chains, which are drawn in orange. And then these different parts of the molecule still have different functions, and we know what they're doing there. For example, the business part of the antibodies the business end here is where the antibody can actually bind an antigen or a pathogen like a virus, or what we are thinking in this case, the prion. And as you see, each antibody has actually two of these ends, made out of a part of the heavy chain and made of part of the light chain. Now, this is a schematic, what the antibody looks like if you were to look with a very high-powered microscope, for example, an electron microscope. You could see something like this. And now on the antibodies, so circled in red, this is the business end of the antibodies. Again, we have two of them for one antibody. And the other thing which I want to mention, what I'm showing you here is just the most common form of an antibody, for example, that you all develop when you got your COVID shots. This is the antibodies, the IgGs that defend you against the virus. There's other types of antibodies, and some of them much more complex, but I'm not going to talk about them today because they don't play a major role for our study either. And so I leave that one out for brevity reasons. Now, these were the natural antibodies, and obviously we now these days we can manufacture antibody fragments in different forms. For example, we can just manipulate the binding site. So here you have the binding end on the Fab fragment on the left-hand side, and it just is a piece of the heavy chain and a piece of the light chain, and it's still stuck together so it binds. But we can also produce these antibodies or their fragments in bacteria as what we call recombinant proteins. And here I'm showing you a single chain variable fragment, which still has the binding ability that is imbued in the Fab fragment and the full antibody, but it's now just one chain, so it's easier to manufacture. And there's also the thing when you just use the light chain part, it still can bind to the antigen, and we call this one a nanobody. And this one is also produced in bacteria. And actually, my graduate student, Vinit Rathod, is producing these things in my lab. And here you see, for example, the more complex view as structural biologists like to show, where we see this, it's called a ribbon diagram, see the bits and pieces, and you can see the Fab fragment, of course, on the left-hand side is a rather complex molecule. The single chain variable fragment is much simpler, but you can see the two different parts that are there that are attached with a linker, and then the nanobody being much smaller. And all of these could act as therapeutical agents, and we would like to test them in our experiments as well to see if one or the other is better in acting there. And there's also other technical details that we're looking at for this study. Now, immunotherapy, we're talking about the prion diseases here, and as most of you will be aware of, we have two different forms of the prion protein to deal with. One is, of course, the healthy form of the prion protein called PRPC. This is the protein we have on all our nerve cells, white blood cells, and it has some function that we don't fully understand, but it's not causing disease in this form by itself. So this is the healthy form of the protein. And again, I show you this ribbon diagram, and I show you, again, surface view of this protein, what it looks like. And we understand that one pretty well. On the right-hand side is what I've shown is PP scrapie, or this is a disease-causing, so the infectious form of the molecule. And it is only a model. So this is not a real experimental structure, but it has a lot of benefits for it, and I'll come back to this one in a moment. And both of these could be targets for immunotherapy. Now, immunotherapy, of course, has been tried before, trying target the cellular prion protein, the healthy form, and the protein being reasonably large, so the antibodies can target different areas, and each antibody will target just one area of it, and we call these areas epitopes. And so there could be different antibodies targeting the cellular prion protein. And in fact, there was this one antibody, PRN100, which has been given to 6 CJD patients in London. The latest news release is from 2019. We have not heard anything further. So it's unclear to what degree this antibody may have given benefits to the patients. And I do not want to speculate on it, but given that it's been silenced for so long, I think it has not given that much of a benefit to the patients after all. Now, and this is our idea, and antibodies, of course, can then target the disease-causing form of the protein. You need to have some understanding about what this one is. And that's exactly the problem why nobody has been able to even try this one in a concerted way, because nobody had a really good antibody that was specific just for the disease-causing form of the protein. And that, of course, you need to have in order to do this kind of trial. And this is where our project really took off, because that's what we developed. Now, this comes from our other project that we have, also CJD Foundation supported. And so this is a project for a prion vaccine. And our prion vaccine also came back from this model that he had. We had selected these surface expose parts of the protein shown here in green and cyan, that in the structure or the model of P. episcrapi, the infectious prion protein are coming together and form again what we call this epitope, so a target for a vaccine. While on the cellular protein, prion protein, these residues are scattered all over, so they're not coming together. The immune system doesn't see them together, so they're not. This was a part of our vaccine project, the starting point. And I have shown this data before, so we tested this vaccine in a transgenic mouse model of GSS, and these mice were given to us by Dr. Glenn Telling from Colorado State University. Now these mice, because they carry this human mutation, they do develop disease. It's shown in the green curve here at 177 days, so that's when they start to get sick, and this is because they carry the mutation, so this is a mutated form. If we give these very same mice a dummy vaccine, so a control shown here in brown, and they do not get any benefit from it. Now, we were very happy and still are very happy about what happens if we give them our vaccine that we've developed and that we're still developing and testing. And this is shown in the blue curve. Now, these very same mice, they stay healthy until about 450 days of age. So this is very great. And this is, of course, the key of our vaccine project. Now, what is going on? Why are these mice staying healthy so long? As you know, a vaccine induces antibody. And here, these the black triangles shows you when these mice were immunized, and we just gave them the vaccine at this stage. And of course, the immune system developed powerful antibodies. And these antibodies are then present throughout their life until they start to finally succumb to the disease. So we know these antibodies play part of the immune role. They are not the only part. There's also immune cells that can defend against it. And actually, we are right now studying which part of these is more dominant or plays what role for the efficacy of this vaccine. Now, what we had is, of course, we had mice that make these antibodies. And so we said, we know these antibodies can protect these mice against the GSS prions that develop in this transgenic mouse model. Now, can we do something with it? Now, when you have mice that make antibodies, you can make what we call monoclonal antibodies, meaning you make them just from one cell. You really go into the spleen, harvest the spleen from a mouse and get this one out. And we got some monoclonal antibodies there. And we found that these antibodies recognize only the infectious form of the prion protein, which is very exciting to us because nobody had really had good luck with these antibodies. Some have had some, but they were not as powerful as I believe ours is. Now here you see what the graph shows you what we do in an assay with this antibody. And this antibody, as I show here, can recognize GSS prions, familiar CJD prions, FFI prions, and even variant Creutzfeldt-Jakob disease prions, while a control, and this is a sample from a non-neurologic human brain, and it's not recognized also as a mouse control. These ones are also, these samples are human brain tissue that was donated by the families of the patients who died from this disease. So again, we're very grateful for the support there. And it allowed us to validate that this antibody really is very powerful. It is even so powerful that when we tested it against animal prions, and so we have BSE prions, chronic wasting disease prions, a whole range of them, and you see these curves here, these bars, shows that this antibody recognizes all those prions equally well, while the controls is the same mice or hamsters without the disease, and as you see, they are not recognized. So we knew from that we have a very powerful antibody in our hands, and that, of course, became then the real starting point for our project. Now, this is the model of the prion protein, the P. episcrapi again, showing the epitope that we use for the vaccine. Now we determined that our antibody recognizes just these two bits of the prion protein, just these two parts on the surface, these amino acid histidine and the aspartate there. And that's the site where the antibody binds to it. And that's an antibody that we find essentially in all of these mice. So it's really the immune dominant component of the immune response that we get in the mice when we vaccinate them with our prion vaccine. And so this is the antibody that we are working with for the current project as well. Now here is a table, and you should not read it. It just shows that my graduate student has been very busy making all kinds of different constructs, meaning he has taken the gene from the antibody, from the mice, put them into E. coli expression system, and makes them in bacteria in all kinds of forms. And we want to test these ideally for this project. And so we have these antibody constructs now. We know they're active, and I can show you some here. For example, when the NEAT made this single chain variable fragment antibody construct, so just these two bits together in one, and use the competition assay. So this is a full antibody as it came from the mouse. And when he adds this construct here, you see the reaction goes down because this one is now competing with the full antibody and making that one not work so well in the presence of this one, which shows us that this antibody is as potent as the full-length antibody, which is very important for our experiments. And so this is one of these constructs, and this assay is crucial for us because we need to show that our antibody production that we have produces actually molecules that are active, that can work, because otherwise we wouldn't want to do the lengthy experiments that come next. And this is what I'm showing you here. So now we have a transgenic mouse line, meaning these mice express the human form of the prion protein. They do not express the mouse form of the prion protein, and we can infect them with CJD prions. And we have done this one in my lab. And if we do this one and we don't treat them at all, these mice start to come down with the disease at about 230 days, which is a long time, but this is the best we can do with our mice. And we will use these mice, the ones that have been infected with CJD prions, and then treat them with these various antibody constructs that we have produced, and also a control antibody that has nothing to do with prions. And then we will observe them. Do they develop the disease at the same time? Or ideally, we would see some expansion of the disease before they come down. That would be our aim. Of course, the real aim would be that they don't get the disease at all, but and this is a very high goal, and I'm not sure that we can get there, but obviously it would be our desired outcome. Now, to summarize, so I showed you today that monoclonal antibodies that recognize P. episcopia, the disease-causing form of the protein, is something we produce, and they will be tested in transgenic mice that are infected with CJD prions. Now, the antibodies themselves originated from mice that received our prion vaccine that is based on the structural information that we know. And we created different versions of these P. episcopia specific antibodies and are producing them in E. coli bacteria. And we're testing them under rigorous conditions for the activity, which is very crucial that we do this one first. And then and this is still in our future, then we will inject them into these mice, the mice that express the human prion protein and infected with CJD prion. And then we have to observe and analyze to see what happens next. And so this is a very exciting time because we are getting very close to getting those experiments started. We now have the antibodies almost ready to go. And then, so now we have to get the mice to go soon. And in the end, of course, I have to thank all the people that did the work. And this is our group picture is outdated. I haven't been able to take a group picture in the last 2 years. And particularly Vineet, Andrew, Lily, and Brian were the ones who did the work. But everyone else in the lab contributed. And of course, I have lots of collaborators, and a few of them I mentioned that help with this project in one form or the other. And then, of course, I want to thank again all the funding agencies, the ones that worked earlier. And again, in particular, I would like to thank the CJD Foundation for supporting the vaccine project that I'd mentioned in passing, and particularly working and supporting our antibody project right now, which is very important to us. And I hope this was clear, and I'll be happy to entertain any questions. Thank you.
Well, thank you to all of our three speakers for presenting their work to you, the families, who've obviously funded this work through the CJD Foundation, and for the way they've explained what they're doing, which is obviously highly technical, but hopefully in an accessible way to you, the families. So we've had, broadly speaking, 3 questions submitted by the spreadsheet. The first of these really relates to a question I was going to ask if no one asked any questions. So you've all mentioned in a way that it's possible to treat these diseases by downgrading or removing the normal prion protein. And obviously one of the questions that was put in this spreadsheet was just to confirm that if one did this, it would reduce the disease process, but not have any bad effects. And you've all fairly carefully said that removal or downgrading or knocking out the normal prion protein doesn't do any harm. But this is effectively based on mice. And I just wondered what you really thought, whether you thought that it would be perfectly safe to knock down or remove normal prion protein in human beings over a long period of time who live in the real world, rather than in mice for a short period of time who live in laboratory control settings. I don't know who wants to take that up first.
I can try if you want, Richard.
Okay.
So, of course, it's a tough question because as scientists, we always like to talk by the data. And as you said, there are no data, of course, of since there been no treatment so far that was able to suppress PRPC in a human being. So, but that said, I'm very optimistic about the possibility for several reasons. Well, the first one, as you said, in experimental models, and not just mice, actually, I think has been done also in cows, as far as I know. Then there is this kind of observation, indirect correlation that Eric Minikel and a few other colleagues made about the possibility that at least a couple of people on planet Earth walk with just half of the expression levels, most likely, of the prion protein. And apparently they were completely fine and like 60-ish years old. So collectively, all these data are suggesting that that's definitely a risk, but a risk that, in my opinion, we should afford.
Okay, so I mean, I think that the answer to the questioner was obviously is obviously that there's evidence to suggest that one can do this safely, but we don't actually know for sure. And sometimes surprising things can happen when you move into human practice. Related to sort of that question was obviously, if you're going to, someone asked if you're going to knock down or suppress normal cellular prion protein, who are you going to do this in? And that brings us to a general question, really, as to whether you are going to give this to people who've got established disease, or whether you're going to give this to people who are at risk of developing disease to prevent it. And one of you, I can't remember, did tangentially refer to the PRN 100 antibody experiment, which of course we know nothing about the results of that. But of course, one of the things that's difficult is that if you treat a disease which is already well established, it may be quite difficult to get results, as people have found out in Alzheimer's disease. So all of you who are thinking of these treatments, certainly to suppress PRPC, are you thinking of doing this as a preventative treatment in people at risk or in people who've got established disease? And also along that line, I think the questioner kind of implied in the spreadsheet that maybe it could be given to everybody. If it really is harmless, why don't you suppress it in the whole population? Anyone want to answer that question?
Yeah, I'll leave the stage to perhaps to Jason, and then I'll express my opinion, I guess.
I'll take a quick shot at it just because it's quite a technical question and very multifaceted. So it's very difficult to try to answer that, because while we all kind of talked about the lack of danger in knocking down the natural protein, and that there's animal models that show that they're healthy, long-term studies, as Miano said, aren't really, obviously in humans, aren't done. But when we're talking about long-term knockout studies on the genetic basis, we're talking about almost a completely other field of science. So when we're talking about knockout models in mice, we have scientists who are very, very proficient at creating knockdown mice or breeding them in a certain way. Just to give somebody something that would create a complete system knockout is very, in my opinion at this point, quite beyond our ability of genetic engineering. On the safety side, surely, because while we've seen huge improvements on either genetic transfections or genetic engineering or genetic delivery to try and treat other diseases, to do a complete human knockout on a global scale definitely is quite beyond our scope at the moment. So while even if it would be feasible, and if we showed that it wasn't dangerous and that it would be good, we're branching into a huge other region of genomics that would be quite difficult to even pass through the general population, I think, as there's quite a bit of not bad blood, but it's not a globally accepted field
But to pick out another, because someone asked a question similar to that actually, and that really does relate to the fact, which I have to say I myself am rather cautious about. If you take the classic disease, which most prion diseases are in the end, statistically, it's sporadic CJD, which is a very rapidly progressive disease in the middle-aged and elderly, usually. And this is a disease which, by the time it's diagnosed, has caused quite a lot of havoc in the brain already. So I guess what I'm really trying to get from you specifically is, do you think that this treatment that you propose is more likely to be a treatment in people who are very definitely at risk, like, for example, carriers of mutations? Or do you think you really have a chance of treating an established disease like sporadic CJD?
I'll try to answer this. That's always the most difficult question to answer, I guess. As a general perspective, the later the worse, meaning that we need a very early diagnosis. We are all aware of that. Not just, for any treatment, not just the treatment that that is trying to suppress PRPC, but any treatment will need a very early diagnosis. So a diagnostic tool will be as important as a therapeutic tool. That's out of question. That said, pros and cons, meaning that a long-term treatment of a person that will eventually develop the disease, and of course I'm talking about the genetic cases, will expose the person to that risk that you were referring before, Richard, and also to whatever side effects the drug will have. We know that there are no drugs with zero side effects. There are drugs that are well tolerated, but long-term treatments are always an issue. But of course, in that regard, the drug will have the maximal chance of being active and producing an effect. On the opposite side, it will be very challenging to get into a person that already developed severe phenotypes. And we know that this disease, and in many cases, is very, as soon as it appears, is very aggressive and rapid. So in that regard, again, it will be very kind of challenging for any treatment. I can only refer to surprising data that, for example, Giovanna Malucci reported, a decade ago, that even when, of course, still I'm talking about the mice, and a mouse is not a human being, but that's we take what we get. In animal models, it seems that effectively, if you reduce PRP, and I'm not talking about actually abrogating completely the expression, so we can even play with levels and, of course, dosage of eventually of the drug that is causing that partial suppression. Apparently, in the mice, it's actually a viable strategy even after the onset of the disease. So the only parallel that a scientist can make is that if we believe that the disease in the animal is recapitulating the disease in the human being, then there is hope. And that's all I can say, I guess.
I think that's a bit of a paradox that you pointed to, though, that, if someone's perfectly healthy before they develop disease, then the safety of the drug is paramount, of course. On the other hand, if they've already developed disease, you're less worried about safety, but you've got less efficacy. So there's a question to Dr. Villa, and that was, your mice live longer, but they died. So was that because the antibody faded away? And could you give booster vaccinations, was the question, to keep them alive longer? And I suppose underlying that question is, why did they die?
Yeah, so the mice died because they were making the mutated form of the prion protein, the GSS prion protein, throughout their life, and they were overexpressing it on top of that one. So throughout their life, they were making it. We did actually measure the antibody titers in their blood after the immunization, and when we had to euthanize them at the end, and the antibody titer had been reduced by a factor of 100. And that's normal for two things. These are now elderly mice, and we know, unfortunately, in the elderly, the immune system is weakening. We all know this one from our own experience. And yes, we only immunized them at the very beginning with our shots there, and then on purpose did not give them any booster because we wanted to find out, do we even have an effect at all? So we started another experiment where we give them more boosters, It's not fully analyzed yet. It doesn't look too good. So we have to think about, is our antigen, and of course we give them our vaccine together with an adjuvant. Is this getting too toxic to the mice? We have to analyze what's going on with these mice. We did not see the extension in the degree that we wanted to see there. I still thought it was a good idea to give these boosters. I mean, as you know, we are all looking if should we get a COVID vaccine booster or not. All these kind of questions are about. So we have the exact same questions for our mice. Should we give them a booster? I was not happy with the results. Now we have to analyze what's going on. Why did we not see a real extension in the phenotype that we see? But again, what we have seen, of course, is very nice. We are very happy with this one. And of course, we're trying this one in many other additional mice that I didn't have time to talk about.
Okay, there's another question for you, which mirrors one of my own questions, although my version of the question may just reflect my poor understanding. And the question is, Dr. Villa, as I understand it, the PRP-SC can fold in different ways. So will your antibody work for all the different ways of folding? And I guess that's what underlied my question, really. That is, you talked about antibodies to PRP-C, which seems fairly straightforward. But then you talked about antibodies to PRP-SC. And then I think you talked about prions, and you talked about the infectious form of the prion protein. I mean, my understanding, which may be wrong, is that, of course, in the process of converting to what we find in aggregated amyloid forms, the prion protein goes through various intermediate forms. And I'm not sure whether your antibody is to the final sort of aggregated amyloid forming protein or to some other prion protein form on the way.
Yeah, that is a very apt question. And we're looking in this one, and there are some interesting studies. I know Emiliano showed a new structure that was published from Alison Krauss and Byron Coey, who also have talked here in the past in the CGD Family Foundation conference. And they have a structure. And interestingly enough, our antibody would not recognize the structure that they have published. In our experiments, all the prion samples from infected animals, for affected humans, our antibody recognized them all while not recognizing normal brain homogeneous. So whatever our antibody is recognizing is present in all of these disease forms that we could get our hands on, which is quite a few. It is apparently not the final structure that Alison and Byron have published there, because that one is completely incompatible with the epitope that we have characterized. So this is a conundrum for us. What's our antibody binding? Is it the infectious form? Is it intermediate? After all, this antibody came from mice that were immunized with this vaccine, and we've seen what the vaccine does. So it does something good to those mice, at least, which is good. Needs to be developed more. And that's why we thought this antibody had a great chance in this project. Apparently the reviewers agreed to give us the money so we can try it. But now we have the question what it is really binding to. That is really a good question, but we can see it in our experiments. It's doing something in the mice, and it's binding the samples in the right way. There's still some questions we cannot answer.
Okay. There aren't any other questions in the section, so I thought I would ask you each in turn a particular question, because I'm simply a clinician, so I deal with patients who have obviously symptoms and disease. And it's clear to me that however interesting the protein pathology is, people become ill, they develop symptoms and they die because neurons become ill and they die. So the critical question, at least to me, is why do those neurons become ill? Why do they die? What aspect of this protein pathology is making these neurons become ill and die? Is it what you see deposited in the tissue as amyloid or something else? So I wondered if each of you in turn could maybe talk about that, and alongside that, what it implies for your treatment. So Dr. Biasini, do you want to go first?
Yeah, sure. Again, $1 million question, meaning that at the moment, I don't think that no one on planet Earth can safely robustly answer that question, because the reality is that we still don't know how prions kill nerve cells. Now, I've been working on a specific hypothesis that is somehow reshaping the kind of ideas that historically people had about prion diseases. And it's the idea that the normal form of the prion protein, again, PRPC, the one that we all have on the surface of the cells, is not only acting as a substrate for the replication of infectious prions, but somehow at the very beginning of the contact between the infectious particle and the protein, the normal protein on the surface, some sort of toxic signal arises from that contact and eventually leads to neuronal death. Now, what I'm talking about is a lot of, let's say, indirect evidence that point out to this possible mechanism. And surprisingly, these possible mechanisms as something to share also with a possible role of the cellular prion protein with other diseases like Alzheimer's disease. That's the work of Steve Striedmatter and a decade ago. So we're still trying to really nail down the real role of PRPC in this disease. And so my only question, my only answer is that our entire conceivement of a therapeutic treatment all the possible directions point out to PRPC as the most likely target. And I'm going to be honest, I've been trying to target the normal form of PRP with many different strategies back to, again, 10 years ago. And not only me, but me, my collaborators, and other labs as well have somehow failed so far to target with canonical classical pharmacology PRPC. That's why we don't have at the moment a good drug to deliver to human beings that really, at least, if not suppressing, but at least blocking whatever activity PRPC is doing. So that's where the project that I presented started, to try to be innovative in that regard and try to stop the folding.
Yeah, so obviously, I'm asking the question about pathogenesis, but as you say, no one really answers that fully. But you've outlined there for people that PRPC may have another role, actually, rather than just simply fueling the production of PRPSC.
I am personally convinced that understanding the physiological function, so the normal function, the biological function of that protein in neurons, will really boost our chances to understand the pathogenesis as well. But that's really incredible that, we know that protein since, beginning of the 90s, and more labs are interested to understand, study, the conformation, whatever. And the function still is kind of elusive in some way. So that's really a mystery to me.
Right. So, you've obviously linked it to why you're targeting your treatment in the way that you do. So, Dr. Dusky, do you want to attempt to answer about pathogenesis and how it relates to your theories on treatment?
Well, I have to mirror most of what Emiliano just said, because he explained most of it very, very well. Expanding it a bit more, he brought up Alzheimer's. We have the tau plaques. We also have a lot of other CNS diseases where you have either aggregation or accumulation of a toxic protein that can no longer be degraded within the lysosomes or can no longer be recycled into the cell. And so these lysosomal storage diseases can kind of mimic that, where we start to see death, because it's just after a certain point, you bog down the system and create problems. That's one of the reasons why, obviously with the molecule we have, we look at knocking down both the PRPC and the PRPSC. But one of the big things we're really shooting for is the CNS delivery. Because if we can get into the brain, not just with our molecule, but if we can get to the brain and show that we can get an effect on this protein, it would open up an avenue for all of the research that we've seen here today that maybe can transport something more simply to the brain and save some of these cells and have an effect, not just with our system, but more on a global scale. But for other lines of research, even the potential of like reactive oxygen species or whatever mechanism that these proteins are causing for the cell death. By no means would it ever be a magic bullet, but obviously to save the cells, we have to get to where they're dying, and that's kind of our major goal in it, is to make sure that we can get to the brain and get into the CNS where we can try and start having a therapeutic effect and saving these cells before they become self-toxic.
Okay. So, Dr. Villa, you've obviously targeting the abnormal protein. I mean, what are your thoughts on pathogenesis and how that relates to your treatment strategy?
Yeah, so there was a great study from Adriano Aguzzi's lab in the 90s when he was first made these knockout mice, and he could show that these mice, if you in various forms bring P. scrapi into the brain of these mice, they do not get the disease, meaning P. scrapi in absence of PRPC is not toxic to the neurons. They do not die. There was no effect on these animals. So this obviously shows that yes, PRPC is a great target if you can get a reduced, removed in various forms for treatment options. Of course, we are looking now if our antibody, for example, as one possibility, binds to the prion protein, the infectious form, the one that's causing the ones, so it cannot bind to the cellular form. If our antibody would interfere there, this could be a pathway to treatment options. There's other ways you could try to do this one. So that's why we are focusing on, based on our work on the prion protein, the infectious form in particular, targeting this way, but we know that the cellular form is crucial for the pathology. Without the cellular form, there is no pathology. And so, yes, it gives us the advantage that we have two different targets that we can try both separately. We could even envision, as you know, in AIDS, the virus, people targeted with various different medicines, targeting the different systems, and have great success with this. So in the end, if somebody tells me 5 years, or there's optimistic 10 years or 20 years from now, we have medicines that target both PRPC and PRPSC, and that helps this one, I'd be perfectly happy with this one. And I definitely see that both of these pathways could lead to success and maybe prevent some of the pathology of happening.
Well, you've neatly picked up the last couple of questions in a way that I was going to present to you, because I think we've got a few minutes left. And that was, you're obviously outlining different ideas of treatment here. And as you've, I think, just really implied, Dr. Villa, I mean, I'm interested to know whether, if telling the families, whether you envisage that there will be a treatment that eventually emerges, as it were, from all the competition, or whether we're looking probably at having to treat the disease in multiple different ways, you know, giving maybe 2 or 3 combined treatments for each person. Do you want to answer that question in reverse order, starting with you, Dr. Villa? But I think you've already given the answer, probably.
Mm-hmm. Yeah. So there is, in medicine, there is this trend called precision medicine, meaning that we analyze our patients in much more detail than we were before with our molecular tools that we have these days, and figure out, can we make a better medicine by targeting the specific forms that this patient is showing us. The same is happening in development for Alzheimer's disease and Parkinson's disease. We are looking into these things. And if in some years from now, we may have these tools that we can diagnose it more precisely to seeing which form of the prion protein, the diseased form, is causing the disease here, because we know there's more than one form behind it, at least subtle differences exist. And if we had specific medicine that would target one versus the other, that may actually help us to really prevent the disease or stop it in some form or another. So I can definitely see that this approach of precision medicine, and of course it means much more precise diagnostics are needed, and this is more development on the diagnostic side that is required. But I could see that there is a group of treatments, and it needs to be targeted to what exactly this patient is suffering from. We know from the genetic diseases they are relatively uniform within one mutation. If you carry of this mutation versus that mutation, that already helps us to understand some of these things. So we are also working with some of these mutations in the mouse models and targeting them. We see some success there, and it may help us to develop different treatment options for one versus the other, and we have to see to what degree they would be applicable to sporadic CJD, which, as you said, is the most difficult one because it's so rapidly progressing and it's usually diagnosed pretty late in the game. Again, diagnostic improvements there are needed that we even stand a chance.
Yeah, I mean, I think just picking up on that, one of the difficulties about sporadic CJD is by the time someone develops the very first symptom, they've probably got very serious established disease. That's so early diagnosis, you almost need early pre-diagnosis, really. Mm-hmm. So you've obviously talked about having different treatments, as it were, for different types of disease, or if we classify the disease as the different types, we might have different treatments targeted for each type. I just wondered, Dr. Dusky, whether you felt that in the end we might be treating everybody with drugs, more than one drug, to attack more than one mechanism, as it were, rather than just deciding that one mechanism might be okay for an individual patient.
I mean, that's kind of the dogma in it all is, do we try the shotgun approach and hope it hits everything, or do we try to be very specific, one at a time? And obviously, one of the major things in that idea is how progressed is the disease. I mean, as you just said, the diagnosis is the first thing we have to look at, because the earlier we can start diagnosing this, the more chances we have to be more specific for one route versus the shotgun approach that we just try to hit everything all at once. While I very, very much like the idea of personalized medicines, and I think that it will be very important in our future, I think it's a little bit ways off because, one, for the diseases where we don't have huge diagnostic capabilities, but also because, as with the antibodies or with our system or with any of the systems we've really seen, if we need to find all of the pathways at the same time, because if we have a really good diagnostic method, but we can only treat one or two of the different phenotypes or the different disease proteins, then we're still stuck. So at that point, then we go with the global knockdown and we try to just completely knock down all of the protein possible and see if that can help it. So while the personalized medicine is very, very interesting, and I believe that we will come a long ways in the near future in trying to strive towards something like that, for the time being, I believe there's a very strong push also towards trying to optimize, so the other side of it, not just the specific for that protein, but having something that will be more, what's the term I'm looking for? Not completely like a global knockdown that would knock down anything, but a much more varied system where small changes in your molecule or small changes in your system will be tunable to try to adapt to different forms, where we can start to try and treat different forms, not everything all at once, but the 2 or 3 major components. And then at that point, if it doesn't work, or if it does work, to get more specific, and these two streets will have to catch up to each other, where as we get better drugs, we'll have to get better diagnostics. As we get better diagnostics, we'll have to be more specific with our drugs. So it's a very double-edged sword.
So before I turn to you, Dr. Biasini, to answer the same question, something's been typed in this spreadsheet, and I think the answer to this, it probably comes from what we've just been talking about, that the disease process might be developing sometime before the first symptom arises. And the question is, how long does the disease process develop under the surface before there are symptoms? And I think the answer to that is, in sporadic CJD in humans, we simply don't know that at all. But the evidence from when we can study these diseases in control settings is that the protein changes and pathology start a fair time before there's actually sufficient neuronal damage for there to be symptoms. But in sporadic CJD in humans, we just don't know. It may be something that goes on for quite a long time or quite a short time. In Alzheimer's disease, there is good evidence that the disease process may be going on for years before people develop any particular symptoms. So, Dr. Biasini, I guess one of the things I was trying to explore about the multiple treatment business was that if you look at all of the work, and it's not just all the three of you, but lots of people publishing on this, people are identifying different kind of pathways or processes that are involved, and then looking at treatments for their particular pathway or process. Do you think that we're going to have, as it were, treatments that target more than one of these? I mean, sort of multiple process treatments in an individual?
Yeah, I think this is actually the easiest question, because we can take examples of other diseases that are treated these days. And I'm referring, for example, to cancer. Combination therapy, that's what I guess we're talking about. So, attacking the disease from multiple angles, of course, required knowledge, required, robust and rigorous science behind each one of those treatments. But then if you are really successful with multiple treatments, then the chance that you really are much more successful than a single treatment is very high. We're learning from many diseases that, not just that cancer, actually. So my simple answer to that is the more the better. In this regard, this is not a race between labs. We are actually all together trying to deliver the vast majority of our efforts to deliver a treatment, each one of us, I would say, with a different angle. And of course, the more treatment we will have, again, the higher will be the success rate here.
Yeah. So I think we're probably coming towards the end of the time that we were allotted. I mean, the only other question that I had is a terrible question, which I'm sure none of you want me to ask, but still, and that is in terms of the families. I guess what they always want to know is when there will be treatment. And what you've described is obviously very hopeful research, and we need to give families hope, but we obviously have to give families realistic hope. And I just wondered if, again, each in turn, you wanted very briefly just to say, what you thought. I know you haven't got a crystal ball, but do you think we're nearly there, or it's 20 years away, or whatever? I know it's a terrible question. Dr. Villa, do you want to start?
Yeah, so it's a difficult question, particularly since, let me phrase it into two different parts. So for sporadic CJD, this is going to be still a long time off. I know this is the majority of patients who are affected by this disease, but it's also the hardest one for us because we cannot diagnose it early enough. We are still grappling, trying to do anything on mice. This one is really difficult one. For the families who carry one of these prion mutations, there are different developments. Sonja Wallab has talked here in previous occasions, and I recently heard another presentation of her in a different place. So the ASO, these antisense oligonucleotides, seem to be working very well in some of her mouse models. I think our vaccine approach looks very promising, and we have, I don't dare to call it an advantage, because if you carry the mutation, of course, this is a big problem that is in your future there. But we can then know the knowledge that you have this one allows maybe us to treat earlier. I mean, the ASO could be infused in a carrier before. Same for the vaccine. We would give this one to a patient who is still healthy before any symptoms show up. And that's also a big question, we don't know if our animal studies translate into humans, we might be able to prevent or delay onset of disease there. That's the advantage, if I am allowed to say the word in this context, with such a terrible disease of the familiar forms. Sporadic is really the difficult one that we will be struggling on for much longer.
So, Dr. Dusky.
So I'll kind of expand on that. The research is coming along, as we've seen from the 3 talks tonight with the other groups going on the other nights. And one of the hardest questions always to ask is how soon will a drug come out? Isn't always based on the results that we're seeing in lab either, though. I mean, we always have nice articles that are being published that are very promising and whatnot. And a lot of times we get blocked in the clinical trials or the upscaling, the industrial scale-ups. And in that sense, I can say that from at least the nanomedicine side of it, which is a little bit different, but also from the vaccine side of it, at least we're seeing a lot of hope in that the research that we're doing with positive results will be translatable more readily into something that could end up on the market. Recently, before in the lab, before a couple of years ago, the scale up of a nanomedicine-based anything or the scale up of a nanomedicine-based vaccine was very rare. The cost, the FCC, the FDA approval, all of that was very difficult to reach. With the new vaccines for COVID and whatnot that are being produced on large scale, we're seeing that with industrial backing, that it is potentially possible to be able to make these kinds of treatments that we're seeing in the lab on small scale into something that could be potentially marketable. So we need to keep improving our results in the lab on the small scale, but at least now we're starting to see on an industrial or a global scale that it is possible or more readily possible to be able to convert the results that we're seeing in the lab into larger scale, which is very, very important and was a very big bottleneck previously for any kind of nanoparticle based or vaccine, a lot of vaccine based systems.
Okay, so Dr. Biasini.
Yeah, so at the moment, at the stage at which our project is now, if we keep following the things that at least we know we need to do, it will take minimum between 5 and 10 years to get the molecule, at the level that we can test it into a clinical trial. That's why, but that's the academic speed. There's nothing to do there. That's why I can disclose I'm trying really hard to find to establish partnership with pharma companies, because I think that at the level in which our project is, my best shot will be to partner with someone that can have the shoulders large enough to speed up the process that at this moment is a typical process of drug development. Once you identify the target and define your mechanism and find a hit compound, then at that point we're no different than any other molecule that is developed for human diseases. Of course, with the stringency that we have to hit the brain, like has been said already. So an additional step to overcome. But again, it will be a matter of being lucky that everything will nail down until the end, and not really say anything that would be unfeasible, let's say. So my estimation is between 5 and 10 years. If we're lucky, we'll get it into a clinical trial.
Okay, well, thank you. I mean, it's obviously not immediately around the corner, but on the sort of visible horizon, sort of, I guess you're saying, yeah.
Well, we need to start somewhere, right?
Yeah.
At this point, with no treatments in the clinics, so we need to start somewhere.
Sure, yeah. And I think the difficulty is, of course, that you can get a lot of success with the initial assessment of potential therapy. And then when you put it into human trials, it doesn't always immediately translate. So very difficult. Well, I don't have any other further questions. And I think we've reached the time that we were going to stop. And all of you probably want to have a rest from answering impossible questions. So it remains for me to thank all of our speakers and to thank the CJD Foundation, and of course to thank all of the supporting staff who've put together this Zoom meeting, which we're all gradually getting used to, but hopefully one day we can have a real face-to-face meeting. And thank you very much as well to all of the families who've attended and those of you who put questions forward. And I hope that you've got some kind of answers and some kind of hope from all of this.
Thanks to you, Richard.
Yep. Thank you, Richard.